Hello, and welcome to Learn How Acoustic Liquid Handling Can Improve Your Bioassay Workflows, presented by American Pharmaceutical Review and sponsored by LabSite. My name is Mike Auerbach, Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator during today's event. Today we're going to be discussing how acoustic droplet ejection compares to the standard tip-based liquid handlers. Before we begin, I'd like to inform our viewers that this event will hold a live question and answer session at the end of the presentation. You can submit a question at any time using the Q&A box on your screen. Please also take note of the resource list on the right side of your screen, which contains several documents and links to more information pertinent to this webinar. And now, allow me to introduce today's presenter. Ian Russell has over 15 years of experience in the life sciences industry and the genomics field. He has been with LabSite since May 2017 and currently manages the Echo Liquid Handler product line. Prior to joining LabSite, Ian managed a broad variety of novel technologies, including microarray and qPCR-related instruments and associated consumables. Ian completed his PhD graduate work with a focus on gene expression at the European Molecular Biology Laboratory in Heidelberg, Germany, prior to completing a postdoc at the Massachusetts Institute of Technology in the cell cycle biology field. Before we welcome Dr. Russell to the webinar, we do have two quick polling questions for the audience. So our first question is, have you used acoustic droplet ejection technology before? Your choices are yes or no. We'll give everyone a few moments to choose their answers. And once you've selected your answers, we will now move to our results. So we've got about 40% saying yes, they have used it before, and 60% saying no. I'm sure Dr. Russell will have some comments about that. And now we've got our next polling question. Does your lab utilize Echo Liquid Handler? And again, your choices are yes or no. So please choose your answer. We'll give everyone a few moments to do that. And now move to our results. So currently we've got about 10% who do and about 90% who don't. So we can discuss those a little bit later. So thanks to everyone who has answered. And now we're happy to welcome Dr. Russell to the webinar. Dr. Russell?
Great. Thank you very much for that kind introduction, and thank you to the audience for providing information in terms of the types of liquid handlers that they're using, particularly within the context of acoustic liquid handling. So pleased to hear that a good proportion of you are familiar with acoustic liquid handling, and happy to share with you a little bit more in terms of what LabSite is able to offer around the Echo Liquid Handler, given that the majority of you have not had the opportunity to utilize that instrument, and I think it has some really great benefit. So over the course of the next 45 minutes, what I wanted to touch upon is, first of all, just give you an idea of who LabSite is, and then we'll dive into the technology that underpins the Echo Liquid Handler. How does it work? We'll go into the specific capabilities that the Echo Liquid Handler offers its users. And then we'll touch upon why one would consider an Echo Liquid Handler for bioassay work. And ultimately, when is this solution available to you? So in terms of the corporate history, just very brief overview here. LabSite has been around for quite some time. The company was founded in 2000. And by 2004, launched its first instrument into the marketplace, and that was the very first Echo Liquid Handler. By 2006, we had a number of large pharmas using it in high-throughput screening and asking for greater throughput, and so the launch of the Echo 555 was initiated in that year to address those needs. And much of that early work was focused on samples that were dissolved in DMSO. And over the course of the next 4 or 5 years, the lab site team worked very hard to start develop new processes within the Echo liquid handler that would accommodate aqueous solutions. And by 2011, we released what we call dynamic fluid analysis. And I'll go into quite some detail in terms of what that is because it's quite revolutionary. But it does open up the field of aqueous fluids for our customers. We also launched some automation in the form of the Access Laboratory Workstation in 2011. And then as we saw adoption of Echo liquid handlers into the aqueous field, and most notably genomics, our customers were asking for larger volumes, the ability to transfer larger volumes of fluid faster. And so that inspired the launch of the Echo 525, and I'll go into some details around that. And then just so you're aware, we do have 2 current initiatives ongoing. One is an Echo mass spec program, which is designed to marry the acoustic ejection technology with the loading of a mass spec instrument for highly rapid mass spec analysis of samples. And the other one is a collaboration with some of our external partners, most notably Brooks and AstraZeneca, to develop an acoustic tube. And while I won't be touching upon that today, there will be webinars over the course of the next couple of months that will go into a lot of detail around that. So if that's of interest, please look out for those coming. So I would say 90% of what I'm going to be discussing today really centers around the ECWID liquid handler itself, but I do want to point out that it's part of a larger system solution that LabSite offers. So we have the Echo liquid handler at the core, but around that it can be integrated into an automated workstation, which enables various devices to be integrated with the Echo and plates to be automated within that particular ecosystem. There is a whole series of different consumables that we offer from 384 to 1536 with various coatings, et cetera, to meet a variety of different application needs. And then, of course, software that is available and aligned with the various applications that our customers tend to be interested in. And while we won't go into great detail there, you will get a flavor of what's available around that. The Echo liquid handlers themselves, I alluded to, come in really 2 different flavors. One is what we call the 55X, or that constitutes either the 555 or the 550, and is ejecting a droplet of 2.5 nanoliters. So this is ideal for your high throughput types of scenarios, for smaller reaction sizes, typically in the 0.05 to 1 microliter range, and supports both DMSO solutions as well as aqueous. And then we have the Echo 525, which has a larger droplet size, 10-fold larger at 25 nanoliters, and as a consequence of that, a faster flow rate, and thus is ideal for larger reactions, ideally between 200 nanoliters and 10 microliters. And we see marries well with a lot of the applications that our genomics customers are doing. That particular instrument supports aqueous solutions only. So let's get into the technology itself. So what we're doing here is moving liquids with sound. And at the core of the instrument is an acoustic transducer, which is depicted there in the cartoon on the right-hand side of the slide, right at the very bottom. That acoustic transducer has an acoustic lens built into it, which focuses sound energy emitted from the transducer into the solution, which is contained within a standard microtiter plate. So the gray area above the transducer is a depiction of that microtiter plate. We're showing 4 wells there. The liquid within those is at various heights. That's not a problem for the instrument. And then between the transducer and the bottom of the plate is a coupling fluid, and we simply use water for that. And that simply enables the acoustic energy to travel from the transducer into the plate. And through the focusing of that acoustic energy at the surface of the liquid, we can very precisely and very accurately eject either 2.5 nanoliter droplets or 25 nanoliter droplets, dependent on the instrument, as I described in the previous slide. And this has enabled our customers to really revolutionize the way that they do science, both by reducing the volumes of the reactions they do and of course, the costs associated with those, and to perhaps in many cases, more importantly, improve the accuracy and precision of those assays. It eliminates any washing or pipette tips, and I'll go into some detail in terms of what the benefits of that are, and eliminates any potential for cross-contamination that a tip-based liquid handler always has risk of. So hopefully, in the next slide here, we'll be able to provide video. There we go. So this is the instrument in action. What you see is the transducer moving across the bottom of the plate. It's going to dwell under a particular well. Now it's emitting that light, that sound energy. That sound energy is focused on the surface of the fluid there, and it's ejecting a droplet out of that. We can position that transducer under any of those wells, and then that droplet is traveling to a plate. You can see it in this particular view right here that's positioned above it. So that plate is inverted, and it captures that droplet as it travels across the space between those plates. The plates, as I mentioned before, can be 384 well plates. They can be 1536 well plates. We can transfer from 384 into 1536, from 1536 into 384, all the different combinations there. So really one of the big benefits of the Echo instrument over a tip-based system is its ability to transfer from any well into any well. Now, let me see if I can get to this next video here. Here we go. So not only is it moving under the plate and ejecting those liquid droplets, but there is a step that occurs before that particular point in the process, and that essentially uses standard sonar or medical ultrasound to interrogate the fluid that's in there. So what you're looking at in this video is a situation where the transducer is emitting a low energy pulse into the well. We get reflections from the bottom of the plate, we get reflection from the bottom of the well, and we get a reflection from the top surface of the liquid. So that is the meniscus reflection. And through analyzing the strength of those reflections as well as the time differences between those reflections, we can start to determine a variety of different characteristics of the fluid that's contained within that well, including the surface level. So the instrument can easily adjust to having wells of different fill volumes, as well as some of the characteristics of the fluid that requires tuning of the acoustic energy power. And so in doing so, we eliminate essentially all operator intervention here. There are no calibrations that are needed with regards to the user. When you set up the experiment, you're going to define a specific fluid class, and those are very general. It's going to be buffer or buffer with surfactant, or buffer with glycerol, for example, or DMSO. Very broad range fluid classes, and then the instrument will use this dynamic fluid analysis to determine the other characteristics necessary to ensure that the droplets being ejected are of the correct volume and are highly precise. So the true advantages that our customers have come to expect from the Echo is really falls into 4 different buckets. The first one is in maximizing lab budget through that assay miniaturization, elimination of the tip costs that are associated with a standard tip-based liquid handler, and also elimination of workflow steps. There is certainly increased data quality. Even though they are miniaturizing their assays, very often the quality of the data exceeds what they were achieving before. We eliminate those pipetting variances through carryover from the tips. We don't lose sample on the tips, and I'll show you an example of how that might impact the type of research that you're doing. There's no potential for cross-reactivity. And of course, anytime you have plastics touching your sample, you're always opening yourselves up for bioactive leachates to enter that sample. And through the elimination of the tips, that's just one additional area that you eliminate there. The system is extremely rapid. So the videos that you saw before were slowed down quite a bit. So what it enables you to do is very easily design, test, and optimize your assays in a rapid fashion. It will support using multiple reagent combinations in a single plate, and I'll show you a couple of examples of that. And then it also enables the transfer of different fluid types from a single plate. So because of that dynamic fluid analysis, we can accommodate a variety of different fluid types within a single plate. And then lastly, the ability to automate what you're doing, and that's going to increase the ultimate precision and accuracy and enables a lot of flexibility through that transfer from any well to any well. So let's talk a little bit about what those capabilities enable you to do within the context of your research. So here's an example of a pooling type of application. We've got on the left-hand side what we would refer to as a source plate. So that is the fluids that we are transferring, and on the right-hand side is what we would refer to as a destination plate. So we're transferring fluids from the left into the destination plate on the right. Because we are able to move that transducer underneath any of the wells on that source plate on the left and position any of the wells in that destination plate on the right, we can transfer from any well into any well. And what you're seeing here is an example of 7 different components here that are all being pooled into the wells in that destination plate. So we're able to do many to one, as in this example. We're also able to do one to many, and that may be a situation where you have precious samples or reagents in the context of, say a qPCR reaction or a design of experiment or DOE. And I'll touch upon that in a little bit more detail in this next slide. So here's a typical example of a design of experiment where the goal of the study is to test all of the various different combinations of the components and apply some statistics to select the concentrations across all those components that give the best outcome. And so this particular example here is combining a set of target cells with a set of effector cells. And this is very easy for the Echo to do. And you can see that what this particular protocol has done is taken all of the ABCs from that top plate, arrayed that in the first 3 columns, and then, arrayed those across columns 1 through 9, and then overlaid on that the 1, 2, 3 columns of the effector cells in all of the different to yield all of the different combinations that are possible. Now, this can be done, as was shown in that previous slide, as a standard block design. But I think some additional capability within the Echo really enables some of the biases that one might see in those types of block designs, where perhaps there's an edge bias on either of the sides, to be able to completely randomize the distribution of those samples. So we're looking at the same combinations that we have on the left, but what the echo has done is, or the user has done, is randomize where the echo has put those within the context of the plate, and therefore removed any edge bias that may confound the data analysis downstream. Okay, I'm going to change gears just a little bit here and start going into some of the applications that we see our customers using it for. And in this particular example, this is an alpha-LISA assay looking at TNF-alpha production. And so this may be very familiar to a number of you, where cells are being activated with lipopolysaccharin, and there are inhibitors that are being applied, and then fluorescence is being measured to determine the extent of the inhibition. And typically, this type of assay is done at a 50 microliter reaction volume using 5 microliters of analyte. What this particular customer was able to do using acoustic droplet ejection is to reduce that volume, the total volume of the reaction, down to 2 microliters, and save both in terms of the acceptor in the donor beads, so there's a 10-fold saving there, but also, just as importantly, save on the amount of analyte, so going from 5 microliters down to 1.6 microliters, and get data that is as good, if not better, than the standard 50 microliter reaction. And that holds true across a variety of different applications. Here I'm just showing you a smattering of what our customers are using the echo for. We've got PCR or qPCR types of reactions where a 6-fold reduction in volume is fairly typical there. NGS library prep, customers have seen an enormous ability to reduce up to 100x for that particular application. Sanger sequencing, MDA setup, and some of the SynBio areas, Gibson and Golden Gate DNA assemblies, again, quite an extensive reduction in volume, which flows through to cost savings at the end of the day for those particular applications. For those of you who are doing pharmacokinetic or pharmacodynamic studies, we actually do have an app note around this, and customers using the echo have been able to reduce the amount of input blood necessary for their downstream assays. So they've been able to take it down to as little as 1 microliter of plasma to be able to conduct those studies. And the little cartoon that's on the slide here sort of depicts in pink a typical approach to this, where each of the data points that the researcher is taking requires a mouse, and that mouse must be sacrificed in order to get sufficient material for assaying. Once you've miniaturized that assay and using the echo, now you can start to think of the mouse as a longitudinal yielding a set of longitudinal data sets. So for each mouse, the researcher can dose that mouse, take a tail vein bleed from that. The mouse remains alive, wait whatever amount of time across that time course, take their next sample. And so ultimately what we've seen in some instances is going from the type of study where they're using 36 mice to get all the data points they want, where they're all sacrificed, down to 4 mice total. And those mice, even at the end of the experiment, are still alive. So it removes a lot of that animal-to-animal variability that one would get when each of the time courses come from a different time point data points come from a different mouse. And avoids a lot of the stress-related artifacts that might be associated with these types of studies. And then for those of you using compounds that are quite costly, you can get away with using 75% less compound in that context, since you're only dosing 4 mice as opposed to 36 mice. So it's a very powerful model to put into place there. and we refer to it as microsampling. Changing gears again, just want to touch upon some of the advantages of non-contact dispensing over the standard tip-based liquid handling. This is a case study that is actually coming from a peer-reviewed publication. It was performed at MedImmune. The reference is there if you'd like to look it up. There's of course, many more data points within that. But what I've done is just pulled out just one of the figures from that particular study. And they are looking at the performance of a peptide. These are standard dose response curves. So on the right-hand side of each of those graphs is high concentration, and on the left-hand side is low concentration. And they're diluting down that peptide as you move from right to left. And what one would expect is that you would see the same level of activation that it would correlate with peptide concentration. And what, in fact, this set of researchers observed was, in fact, that wasn't the case when using a tip-based system. So you can see what is shown on the left-hand side in panel A is a constant amount of peptide, but what they're doing is titrating in this case, BSA, so additional protein. And that BSA, it functions as a blocker, and the EC50 of the peptide actually changes as a consequence of the blocker concentration, because the amount of peptide that is adhering to the tips as you do that transfer is changing. Whereas if you do that in a non-contact form, as on the echo, what you see is that the EC50 for that peptide remains unchanged, irrespective of the amount of blocker that's contained within that sample. So very powerful in terms of ensuring that if you are determining you're putting X amount of compound or sample into that sample, all of that is getting in there, and you're not losing material on the outside or the inside of the tips that you're using for that transfer. An additional powerful piece that can be brought to bear here is, of course, I'm assuming many of you are familiar with doing dilutions. And typically the way one would do that is depicted on the left-hand side of the slide here. You start with a concentrated sample. You take a portion of that. You dilute it in that first dark circle. You mix that up, and then you move a portion of that again into a subsequent dilution. And so you serially dilute as you move from left to right. And of course, any mistakes or errors in that dilution that occur on the left-hand side are propagated as you move across to the right-hand side. Now, when you have a instrument that is capable of transferring such small volumes as the echo, you can start to think about doing things like what we call direct dilution. So using the same example, we're starting with that high concentration sample contained within the box, and we're taking smaller and smaller aliquots of that and directly diluting into there. So each of those may have a certain error associated with it, but those errors are not propagated across the dilution series as you make them. And so that's incredibly powerful in terms of increasing the precision of any dose response curves that you may be creating. So the way this would be applied is depicted in this slide here. We have an 11-point dose response curve here. We're starting with a 10 millimolar stock solution, and we're effectively making 3 serial dilutions. So the area on the right-hand side in blue, we're taking the stock solution. We're creating a 7.5 millimolar, a 2.5 millimolar dose point by direct dilution. And then we're creating 2 additional intermediate plates. So we take the 10 millimolar stock solution, we create an intermediate at 0.1 millimolar and an intermediate at 0.001 millimolar. And then we use those again to seed the direct dilution across the yellow data points and the green data points. So the extent of the serial dilution is only one step as opposed to 11 steps, and then all of the other data points are derived through that direct dilution process. Okay, so for the final part of the presentation today, I'm actually going to change gears quite a bit and talk about a new product that we have coming out. So we focused on the Echo instrument, its capabilities, and some of the applications that our customers are using it for. To date, all of this work that I've described to you has occurred in a research environment, and we have customers who are asking us to provide the support and infrastructure to utilize the Echo instrument within a regulated environment, whether that be due to FDA regulations, 21 CFR Part 11, or simply that they want to follow certain GMP or GLP guidelines. And so in order to support these customers, we're in the process of commercializing a new software product which we call Echo 21 CFR 11 Compliance Manager. And what I want to do, sorry, we're one slide ahead. What I want to do here is share with you some of the benefits of that, how that would integrate into your workflow on the Echo, and ultimately, what that solution would look like. So as I mentioned, it is designed to help laboratories achieve FDA 21 CFR Part 11 compliance through tracking of protocols. It enables new protocols to be created, existing protocols to be edited, but through an approval process. It also manages user access to the instrument. It controls who can run specific protocols, and who has access to data, and then finally run log auditing so that any of the data that's generated by the Echo instrument can be easily accessed, and more importantly, is locked down and cannot be changed once it is written to the database. And I'll talk about that in a little bit more detail in a later slide. It is tightly integrated with the existing Echo software applications, and so we'll talk a little bit about what those are. And as I mentioned, it's ideal for our regulated GLP or GMP customers and the types of environments that they work in there. So what is it? What does it do? It is actually two different applications. One is the main application, which enables the management of protocols and the ability to run those on the instruments. And then a second application that we would envisage being used by the IT organization within your company to manage the users, create databases, manage projects, and ultimately define who does and who does not have access to particular functionalities on the Echo itself. So let's go into a little bit more detail there. So here's the software architecture at a very high level. So the Echo itself runs a server. That server communicates with our application software through an application programming interface, or API. I'm showing you a couple of the applications that we have here. I think some of the more common ones: Echo dose response, Echo plate reformat, and Echo combination screen. In the absence of ECM, the user would directly access those applications. Within the context of ECM, when that is enabled on the system, it creates an additional layer that the end user will interact with the system through. And I'll go into a lot more detail in terms of what that enables and how that's managed. That particular layer is associated with a SQL Server database, and actually there will be a number of different databases that enable data to be compartmentalized. So if you are, for example, a CRO, or perhaps you're sharing data with another third party, you can create a specific database to contain that data. You can archive that database and then send it to that partner, and they have the ability to open that up and to review the data that's within it. So what functions does Echo Compliance Manager offer? It really falls into four main buckets here. First one is it sets and manages many of the user permissions. So we can have multiple user groups, and we can partition the data into databases, projects, and studies. It enforces protocol access security. So all of the protocols are stored within that SQL database, and they can only be accessed if a particular user has been set up in a fashion that enables them to access that data. It controls who is able to edit the protocols using the Echo apps. Every time a protocol is edited, it is reversioned, and a new version is created. That old version, though, persists within the system. And then it oversees the runs that are being transferred to the Echo. So only those folks who have access to that functionality will be able to run particular protocols on the Echo. And then it controls the run inputs and the run creation. It also manages the change control and approvals side of things. So anytime a change is made to a protocol, it requires approval, and there is an audit trail for that. And then anytime a run is made, there are a variety of run reports which are output by the Echo itself, which define what the protocol was, what samples were transferred, and whether those samples were transferred successfully. And then lastly, it monitors the hardware and the consumables. So within any run, it monitors what the instrument status is, as well as controlling the instrument labware that the user has access to. The control to the system is done at really 5 different levels. So we have anything from a guest who only has the ability to view any of the approved protocols or run reports. They don't have the ability to run anything or make any edits. We have a standard user. So this might be your analysts within the lab. They are limited to running only approved protocols, and you can define exactly which approved protocols they have access to. We then have one layer up from that, which is the power user. Those particular individuals can do all of the above, but they also have the ability to create and edit protocols, as well as run any of the protocols that are still in a draft form. So that means it's a protocol that has not gone through the approval process. They can also edit any of the approved protocols. And when they do that, that protocol would go back into a draft form and would require approval to be accessed by a user who, remember, only has access to approved protocols. We have an approver who can do all of the above, but they also have the ability to approve any draft protocols that are submitted to them. Now, slightly separately, we have an admin role. We don't foresee that this would be necessarily a role for somebody working with the Echo itself, working within the Bioassay development team or the QC team, but would instead perhaps be somebody within the IT group. They would be able to override certain approval requests. If you have an approver who's left the company, for example, and there are pending approvals waiting there, they can go in and push those through. They have the ability to archive or delete project data. They have the ability to create new databases, to manage the access levels, and to disable any obsolete protocols from being run. The software itself uses a tabulated data organization format, and while I don't expect you to read this, I just wanted to give you a brief flavor for what that might look like within that, both at a database level, a project level, and a run level is what's being viewed here. And then looking at the protocol versioning and the approval process, this is from the context of an approver. They have an additional icon in their ribbon at the top, which enables them to see if they have any particular protocols waiting for approval. They would click on that. They would see a set of protocols and their status. So in this particular example, they're all being shown as approved. If there was one waiting approval, then that status would say draft, and it would have a revision number associated with it. Anytime a protocol is approved, it gets bumped to the next version number. So, for example, in that first one called My Cherry Pick, if I go in and edit that, it would go to, it would stay at version 1, revision 1, so it would be 1.1, submit that for approval. As that gets approved, then the version would go up to version 2. And then lastly, they can dig down into some of the more details around the history of a particular protocol there. As I mentioned, the instrument tracks a lot of information as it goes through a run, and that information is captured in the SQL database itself and can be output from the software in one of two formats. You can do it if you want a format that cannot be changed. You have the option to output that as a PDF. And the nice thing is the system tabulates all of the data for you. Or you can output that as an Excel file if, for instance, there's any downstream analysis that might go into, or whether that might get transferred into a LIMS system, for example. And there, the different reports that the Echo produces are situated on different Excel worksheets. So there's different tabs for each of those reports. It is important to understand that Echo Compliance Manager is a closed system, and this is a requirement under 21 CFR Part 11, and it does meet that particular requirement there. At the heart of Echo Compliance Manager is what we call checksum verification. So we're looking under the hood here at the software. This is actually looking at the SQL tables themselves. And if a researcher was to go in there and tamper with the data that's in the database, here we're changing a particular value. That value has a checksum that is associated with it, that is an amalgamation of all of the information that particular value is made up from. And as soon as you change any component of that information, then the checksum within the database will no longer match the checksum that is generated on the fly. And so you can see what's being done here is to change protocol status from draft to approved. So this actually hasn't gone through approval. This is done external to ECM. And within ECM, when you look at that particular protocol, it is highlighted with that red dot there to tell the user that, hey, something has been changed with this outside of ECM, and the data is no longer correct, and it highlights it for further investigation there. The software applications that are compatible with Echo Compliance Manager are Echo plate reformat. So this particular piece of software enables a broad range of plate transfer functions from plate replication to library compression, formatting, and custom mapping. We have Echo Cherry Pick, which is a very powerful application enabling pick lists to be utilized in guides. Essentially what is a file-driven transfer of samples from any source well to any destination well. So this would be, potentially at the end of an assay, be picking out the positives from that assay. And we have a software specific to echo dose response, so it enables you to very quickly and easily create the different dilutions through that direct dilution process for your IC50s or EC50 types of experiments and generate those dose response curves depending on the number of points that you want and the concentrations that you determine are necessary. And then lastly, for situations where you're looking at the interaction between different drugs potentially, we have a software we call Echo Combination Screen, which enables the easy creation of protocols that not only enable dose response curves for a single compound or drug, but combinations of different drugs to be created within that and do that in a very seamless fashion. So I will, before finishing up here, just quickly summarize. Hopefully what I've shared with you is actually quite a powerful instrument for bioassay development and the execution or deployment of those assays. We've got the ability to do any well to any well acoustic dispensing of any compounds, reagents, samples, and I think this is going to be of particular benefit to the groups that are doing bioassay development and design, just in terms of the amount of flexibility that affords to them. A big differentiator for the Echo is the ability to transfer these nanoliter volume increments with extremely high precision and accuracy. And what we find is even though our customers are reducing or miniaturizing the size of their assays, they are concurrently improving the data quality and the reproducibility that they get from that. And that assay miniaturization is yielding significant cost savings as a result of that. And then lastly, I talked about our Echo 21 CFR 11 Compliance Manager software, which is really taking the Echo to that next level and enables it to be integrated into a regulated environment through the management and creation of the protocols, the approval of the protocols, the fact that all of the information is databased and locked down for auditing purposes. And in terms of timing, so the Echo liquid handlers themselves are available today. So this is a product that's been out in the market for quite some time. What is new is the Echo Compliance Manager software, and we anticipate the release of that software in early 2018. So if you have interest at any or all of these different component pieces, do reach out to your local sales rep, or you can contact LabSite directly. And with that, I am going to stop there and am happy to address any questions that may be out in the audience there. Thank you.
Thanks, Ian. I'd like to remind everyone that you can submit any questions you might have directly into the Q&A box on your screen, and we'll get to as many as we can excuse me, during the Q&A session. So let's get right into some questions right now. And let's take a look at our first question. And Ian, the first question is, can the plate handling for the Echo be automated?
Yes, the short answer to that is yes, it absolutely can. We have automation solutions that we offer through LabSite, and that enables the transfer of standard SBS plates to the Echo instrument and to a variety of different devices that can be integrated into the automation system.
Okay, great. Thank you for your answer for that question. Let's move on to our next one, and that question is, what plates can be used with the Echo? Are all PCR plates compatible with Echo?
Yeah, that is a great question. I didn't go into too much depth around that, so thank you for that. So within the context of the source plate, so this is the plate that we are actually transferring from, we recommend the use of what we call echo-qualified plates, and those are plates that are offered through lab sites. We go to a lot of effort to make sure that the acoustic properties of those plates are consistent across the bottom of the plate so that we do meet those very tight accuracy and precision specifications that we have around the instrument. And those come in a variety of formats. We have 384, we have 1536, we have low dead volume versions of those, and for those of you who might be doing cell work, we have various coatings that can be offered on those. And for those of you that potentially would be working within an automated environment, we do also have the ability to offer barcodes that are pre-applied to the plates according to your specifications.
Thanks, Ian. Let's move on to our next question.
Actually, let me just sorry, Mike. Let me just speak to the destination side because I think that was also a component piece of that question. On the destination side, pretty much any plate can be utilized. It must be of an SBS configuration, but I think most plates today do meet that standard. The question did bring up PCR plates specifically. We do have a requirement for what would be referred to as a hard shell plate. So it does need to be automation compatible, and it does need to have a skirt on that plate. But apart from that, there's actually a pretty broad selection of PCR style plates that are available. And then for many of the other plates, the echo-qualified plates that I just talked about can be used as destinations. But in many instances, our customers are using SBS plates that are specific to the types of assays that they're using quite successfully.
Excellent. Thank you, Ian. Let's move on to our next question. And also, I just want to remind everyone that there's still time to submit a question if you have one. So please go ahead and do so. And our next question is, can the echo transfer cells? Does the sound energy damage the cells in any way?
The echo can transfer cells, and in fact, we do have customers that are using it for that very purpose. The sound energy that is being emitted by the echo is very low energy from the perspective of a cell. So there is no lysis of the cell or perturbation to the cell that we've been able to observe. And I think that also transfers to things like proteins or DNA, which many customers use the echo to transfer. Because that sound energy is so low, we don't see any denaturation of proteins or any damage to DNA. So I think just to summarize, the energy that we're using to eject that droplet is low enough that we wouldn't anticipate that there would be any damage to any of the biological materials that we've seen our customers interested in transferring there.
Great, thank you. Let's get to our next question. Question is there any literature available for utilization of the echo in PK studies, as you mentioned earlier?
I can't say specifically off the top of my head. What I would perhaps provide guidance to there certainly is. There is a great place to start. There was a publication of JALA where that was devoted solely to the echo, and there are a very broad assortment of publications within that particular journal that I believe cover that particular topic. So that would be one place to start. That can be accessed actually through our website. So, given that it was dedicated to the Echo, we did make an arrangement with the publisher to make that available to our customers. So you can actually download any of those papers from the labsite.com website. And you're welcome to contact me offline, and I can go through my database of papers and probably pick out a couple additional ones, or I'm sure your sales rep would be able to provide you with guidance on where to find the specific publications you might be interested in.
Thanks, Ian. Let's get to our next question. The question is, can this instrument handle a solution with less surface tension like methanol?
It can. So we have shown feasibility in transferring things like methanol. The one concern with highly volatile liquids such as methanol is flammability. So we don't at this time recommend the use of the Echo for something like neat methanol. But it is capable of utilizing the dynamic fluid analysis of adjusting the parameters of the energy being used to eject that droplet for a broad assortment of liquids, including very low surface tension liquids such as methanol itself.
Here's another question, possibly in the same ballpark. What types of liquids can the Echo transfer? Does it require calibrating?
Yeah, so it's a very broad spectrum of liquids that our customers are transferring with it. And the way that we enable that is we take on the calibration at this end. So each Echo that goes out the door has a variety of different calibrations on it. And those, as I believe I mentioned earlier, those calibrations are general liquid classes. So we would create a calibration for an aqueous solution, for example, and then we rely on the dynamic fluid analysis to make the fine-tuning necessary to adjust for any differences in that liquid from what we would consider the standard that we use in the calibration process. So we have aqueous, we have calibration specific to solutions containing protein, we have calibrations for DMSO, for solutions containing surfactant, and for more viscous solutions such as those containing glycerol. So glycerol tends to be what we utilize as our standard there. And in each case, we use that dynamic fluid analysis to fine-tune that so that there is no calibration that is done by the user. It's simply select what they believe to be the appropriate calibration, and that's very easy to confirm that it works for a particular fluid, and go. We do also have a broad spectrum calibration, which the crystallography researchers are using, where typically what they'll put in a plate is an extremely diverse set of liquids, and that don't fall into any of those particular fluid classes that I just described to you. And so that particular calibration is able to accommodate that greater diversity, but it does run a little bit slower than if you were to select a particular fluid class. So that would be why you would go for a particular fluid class. If you really didn't know what you had there and what the appropriate one was, you could go for this broad spectrum calibration that spends more time in interrogating the liquid and really dialing in the values for that transfer.
Great. I think we have time for one more question. And that question is, you shared one published example of the Echo being used for dose response curves. Do you have other published examples that I can review?
Yes, there are certainly plenty of published examples that are out there. Just as that previous question, I would encourage anyone who's interested in that to go to that JALA publication as a great starting point. I know for sure that there's additional dose response studies that are published within that. And again, if you're not finding what you're looking for, feel free to reach out to me directly, or in many cases, your local sales rep can point you in the right direction.
Perfect. Thank you very much. So unfortunately, as I mentioned, that's about all the time we have. We would like to thank Dr. Ian Russell for sharing his knowledge with us, and also offer a special thank you to LabSite for sponsoring today's event. Also, keep a lookout for an email containing a link to view this webinar on demand, which would allow you to watch the presentation again or forward it to other colleagues for their viewing. You will also see a survey launch on your screen in just a moment. Take a few seconds to provide your feedback on this webinar. We'd really appreciate it. Once again, thank you all for joining us, and have a great rest of your day.